Good morning, student. In this video, we study the process of fossilization and types of fossil, which belongs to Unit 4, Paleobotany. What are fossils? Remains or vestiges or traces of plants and animals of the past are called fossils. These remains of organisms from past geological age remain preserved in sedimentary rocks either as actual structure or impressions, cast or molds. The word fossil is derived from the Latin word fossils, which means to dig up in the earlier studies. Therefore, a large number of things dug out of earth crust were called fossils. These things also include minerals and rocks besides remains of plants and animals. Later on, however, study of fossils were made restricted to only animal and plants. Study of fossil is of great importance because they furnish evidence of the prehistoric life, as well as they also provide missing links in the evolutionary chain. Study of fossil is now an established science. It has helped in the construction of phylogenetic classification scheme. It has also thrown light on how some of the complex structure of extant plants and animals have evolved. A full-fledged research institute devoted fully to the study of fossil plant now exists in our country. Its name is Birbal Sahani Institute of Paleobotany, which is present in Lucknow. Plant fossils are rarely as well preserved as animal fossils because their tissues normally do not contain calcified structure. They are usually therefore completely decomposed before the process of fossilization. The fossils or remains of a large or macroscopic structure such as leaves, branches, fruits and seeds are called megafossils, while those of very small or microscopic structure, that is spores, pollen grain, etc., these are termed as microfossils. The species of a plant or animals which no longer exist is called extinct. On the other hand, the species which exist at present is called extant. If a fossil cannot be assigned to any genera containing extant species, then its genus is termed as an organ genus. Similarly, if it cannot be assigned to a family, it is placed in a form of a genus. Now, we study the formation of fossils. In the basic process of fossilization, the physical part of any plant or animal must be buried within a well-protective matrix in the crust of the earth. This matrix in the earth crust is usually sedimentary. The sedimentary environment of this kind can be several types such as lake, stream, inland sea or estuarine. In several cases, it has also been observed that diatom fructule also get incorporated in deposits of deep sea basins. In the rare cases, the sedimentary environment is in the form of volcanic deposits or other subaqueous condition. The portion of the organism preserved in sediment become stony or linthified during course of time. How these sedimentary rocks these are formed? Accumulation of rock particles results in the formation of a sedimentary rocks. Weathering and mechanical abrasions of existing rock take place and give rise to the rock particles. Chemical weathering and flooding also help in the formation of these particles. These rock particles or sediment accumulate and water is squeezed out of them. During course of time, this makes them much more compact or rocky structure. Such a rocky structure is called sedimentary rock. There are some conditions which favor fossilization, and these are nothing but anaerobic condition. In that, low pH must be present and forest fire in the form of a fossil charcoal. Presence of sedimentary material such as carbonates, silicates, salt of iron, etc. There are various types of a fossil. First, sedimentary rocks. In them, example is a coal. Majority of plant materials are preserved as a fossil in a sedimentary rock. Coal is the best known example of sedimentary rock. Sediments of plant origin are crushed by overlaying pressure and form coal. Present coal belt in the world therefore represent dense forest of the world of earlier times. List metamorphosed coal shows maximum detail of a fossilized or preserved plant material. Therefore, lignites, early stage of a coal formation is termed as lignite, carry less crushed plant parts and their details can be studied easily. Plant part get excessively crushed in bituminous coal and anthracite coal because they show more degree of metamorphosis than lignite coal. Bituminous coal is the great importance in the study of palynology because pollen grain are best preserved in this type of a coal. Palynology, I told you that it is a special branch in which we study the pollen morphology. That branch is termed as a palynology, and this bituminous coal is of a great importance in the study of a palynology because pollen grains are best preserved in this type of a coal. Then second type is umber. The fossilized plant resin secreted by coniferous trees that grew in every early times is called umber. This every early time in the geological past ranged from Carboniferous, that is about 345 million years ago, to Pleistocene, that is about 2.5 million years ago. Fungal spores, pollen grains were trapped in this resin before fossilization. The resin fossilized into umber and inside these were left spores, pollen grain, etc. Umber is therefore an example of fossil within in fossil. Diatoms are unicellular algae belonging to class Bacillophyceae. Their walls have silicon deposits. The sedimentary rock formed by the remains of diatom is called diatomite. In due course of time, diatoms keep in depositing at the base of sea, ocean or lakes and forms sedimentary rock. Next is pseudofossil or dendrites. Pseudofossil or dendrites are completely inorganic structure of various types. They often resemble plant organs. Their formation takes place by the deposition of minerals due to seepage or pre-collation of water in rock crevices. They superficially resemble leaves of a fern. Next type is mummification. This process of the formation of fossil in ice frozen environments in the polar region is termed as mummification. The moisture of the tissue of the organism gets covered to very small or micro crystals of ice. It is almost a process similar to deep freezing. Next one is biochemical fossils. These are the fossils which consist of chemical substances like chlorophyll, amino acids, aromatic acid, fluorides, branched hydrocarbons, and steroids. These have been reported to be present either in the fossilized remains of organisms or in the rocks. Nicholas in 1981 reported biochemical fossils of the substances related to sporopollenin, lignin, cutin, cellulose, etc. On the basis of the type of fossilization, various names have been assigned to the fossil. Some of them are discussed below. In them, first one is impression. This is a type of fossil in which impression or negative replica of the plant part is clearly visible on the rock upon splitting. The surface of the plant part involved is visible in this type of fossil. The entire shape of the organ is clearly visible in an impression, but cellular details are not visible. During the course of formation of impression type of a fossil, there is no involvement of the organic matter. Second is mold. As the name indicates, molds are formed by three-dimensional structure dissolved by seepage of groundwater, leaving a hollow cavity in the rock. Such hollow cavities resemble with the original organ in size and shape. External feature of the plant in a three-dimensional view are best seen and studied in molds. Example, ornamentation of seeds and fruits. The process of mold formation is similar to modern day sculpture making. A sculptor makes the original with wax or wood and prepares the mold. At the time of preparation of mold, the original statue of wax or wood is removed from inside. The molten material is now poured into the mold to make statue of this material. Exactly in the similar fashion, molds are formed in the conditions when prior to the crushing of a plant part, the sediment surrounding it hardens. During the course of time, the internal plant material dissolves and the hollow of the sediment is the mold. Next one is a cast. Decay of tissue in an organism results in the formation of a hollow. When this hollow gets filled with the mineral matter, that is sediments, it results in the formation of a cast. An exact replica of the original plant material is thus resulted. Like mold, there is also no involvement of the actual part of the plant in a cast. Fruits, hard seeds, and tree trunk are commonly fossilized as a cast. In a cast also, a three-dimensional view of the organ is seen similar to molds. Next, four type compressions. When a bulk of the plant material get compressed in layers of sediment, the fossil are called compressions. By the laying of the additional sediments from above, water squeezes out of the parts of the organism and this makes them more compact and flattened. Ultimately, a thin carbonaceous film remains in the compression fossil and thus corresponds to the original outline of the parts of the organism. Differing from impression type of fossil in which no cellular details can be seen, some cellular details can be seen in a compressions type of fossils. These cellular details include epidermal hairs, cuticularized epidermal cells, spores, etc. If compression type of fossils are formed in a low pressure and low heat, some cellular details, example plasmodesmata, chloroplast with grana, nuclei with chromatin, microfibrillar organization of cell wall, etc. may be observed, although rarely. Much of the organic matter of the plant is preserved in compression type of a fossil. Next one, petrification. Petrification are formed when part of the plant are completely submerged in water reservoirs containing dissolved minerals. In the process of their formation, several types of soluble minerals infiltrate the cells and intercellular spaces, replacing the water and organic molecules. These soluble minerals include carbonates, silicates, and iron compounds. The minerals surrounding the cellular remains precipitate and form the rock matrix. This precipitation is resulted due to gradual evaporation of a moisture. A state of supersaturation is resulted. In this way, a petrified fossil is a mass of a plant tissue infiltrated with the hardened mineral substances so that a large part of the internal structure is preserved. It is like that of a process of embedding the plant material in a paraffin wax in the laboratory. The air and water in tissue and cells are replaced by the impregnating material in a petrification. The cellular details are much better preserved in petrification than in a compression. Next point is nothing but a determination of age of a fossil. By finding the age of a rock, the age of fossil is calculated. The age of a rock is calculated by using radiometric dating technique. In this technique, various radioactive isotopes of uranium, that is uranium 236, uranium 238, thorium, that is uranium 232 and potassium 40 are used. All these radioactive isotopes are called geological clocks. The radioactive isotopes decay and they lead to stable isotopes. In this process, the energy is released. The rate of decaying of any radioactive isotope and its giving rise to stable isotope is always a constant. The age of rock and plant can thus be calculated by measuring relative quantities of radioactive isotope and a stable isotope. The use of radioactive carbon is called carbon dating. Carbon dating technique is used in ascertaining the age of specimen of a plants and animal. Back to about 16,000 years. It is because of the fact that half-life of carbon is 5,568 plus 30 years. Thank you.